right, class, let's continue our discussion on the cell and the structures within the cell. So we've already talked about cell types, cell size, and then different kinds of cell transport with the moving things into and out of the cell through a semi-permeable membrane. This discussion is going to focus mainly on the structures inside of the cell, mainly on the organelles. Firstly, we're going to focus on the endomembrane system. Our endomembrane system is a series of interlinked organelles that all function to make proteins that are preferentially going to be secreted from the cell. Some of these structures include the nucleus, the ribosomes, and our endomembrane system itself. So, first, let's talk about our nucleus. Our nucleus is an organelle that's very large and located in the center of all eukaryotic cells. So all cells that have nuclei are referred to as a eukaryotic cell. If that cell does not have a nucleus, it is a prokaryotic cell, and we have bacterial and archaeal domains that are prokaryotic. The nucleus has a nuclear envelope, which is just a nuclear membrane on the outside. It's a double-membraned organelle or envelope. Inside of the nucleus, we have chromatin. And chromatin is a technical term for DNA loosely bound to protein molecules called histones. So DNA plus protein equals chromatin. We use the proteins in the nucleus to help organize the DNA and keep it from getting getting tangled up, to use a poor analogy. And there's a region in the middle of the nucleus known as the nucleolus. This is a smaller subregion within the nucleus, and this subregion is going to be used to make ribosomes. It contains ribosomal RNA, rRNA. So we have the nucleus as a whole. It has a double membrane nuclear envelope. It has DNA wrapped around proteins called chromatin in it, and then there's a small region on the inside where we make ribosomes. Those ribosomes are the smallest organelle. Our ribosomes are made of both rRNA and protein. So what we notice here is that there's no lipids. The ribosome is one of the only organelles that is not lipid bound. We find the ribosomes free-floating in the cytoplasm of the cell or bound to the endoplasmic reticulum. Ribosomes are important in our cells because they're the little workers, so to speak. They are the little factories that make proteins in our cells, the site of protein synthesis. And proteins are significant because they make up most of the dry mass of our bodies. So here we can see a eukaryotic cell, and we're going to zoom in on the nucleus. Here's our nucleus. The nucleus has double membrane nuclear envelope on the outside. You'll note that there's lots of little holes in the nucleus known as nuclear pores, and here's a scanning electron micrograph of those nuclear pores. Those nuclear pores are significant because they allow information to go in and out of the nucleus. In the center of the nucleus is that nucleolus where the ribosomes are made, and then right next to the nucleus we have the rough endoplasmic reticulum. Each of these little yellow dots represents a ribosome that was made in the nucleolus, and those ribosomes attach to the rough endoplasmic reticulum, abbreviated as the rough ER. From the rough endoplasmic reticulum, the next organelle in the endomembrane system is going to be our Golgi apparatus, which is not shown here. Directly connected to the rough ER is the smooth ER, and the smooth ER is very similar to the rough ER. It does not have the ribosomes attached to it, though. As we look at this endomembrane system, it's the inside membrane, or the inner membrane system. These membranes are going to be involved with making molecules that are transported in the cell and out of the cell. We'll start with the nuclear envelope, the membrane of the nuclear envelope, which is continuous to the membrane of the endoplasmic reticulum. From there, a transport vesicle will send the proteins to the Golgi apparatus where they're processed, and then from the Golgi apparatus, those proteins will be integrated into a lysosome or a secretory vesicle. So here we can see the nucleus, which is part of our endomembrane system. From the nucleus, we're going to go to the rough ER, and the ribosomes will make a protein at the rough ER, and that protein is represented by a blue squiggly line right here. And that blue squiggly line, that protein, is going to be packaged into a transport vesicle, which carries the protein to the Golgi apparatus. We also have the smooth ER over here, and that smooth ER makes lipids, which can be packaged and sent to the Golgi apparatus. At the Golgi apparatus, those lipids and proteins are processed and repackaged. Some of those proteins are going to be packaged and sent through a secretory vesicle to the cell membrane or plasma membrane, where the proteins are going to be secreted via exocytosis. Sometimes those proteins and lipids are packaged up to make a lysosome, which is a digestive organelle, and that lysosome fuses with any incoming vesicles to digest the molecules in that vesicle so they can be used by the body. So in summary, for the parts of the endomembrane system, we have the rough ER, the rough endoplasmic reticulum, which has ribosomes on it and it makes proteins. We have the smooth ER, which doesn't have the ribosomes, but makes lots of carbohydrates and lipids. And then we have the Golgi apparatus, which is used to process and repackage those carbs, those proteins, and those lipids made by the rough and smooth ERs. Lysosomes are a digestive membrane that are made by the Golgi apparatus and are used to digest incoming phagocytotic vesicles or to digest and recycle broken down organelles so they can be reprocessed. And then we have vesicles, which is just a little membranous sac. Just think of it as a travel suitcase that's used to move stuff around the cell. And depending on whether it's going to stay in the cell or leave the cell, generally we'll use the terms transport vesicle for staying in the cell or secretory vesicle for a vesicle that secretes its substance out of the cell. So, concept check, class. Proteins produced in the rough ER would be colloidal in nature, which means that they are going to be easily suspended in solution and not precipitate out. Those proteins would stay in the cytosol. Those proteins may be excreted or secreted from the cell. Or D, those proteins will be modified in the mitochondria. So, go ahead and rewind the video, pause it, and check your notes, or pause it and check the textbook and get me an answer. 5, 4, 3, 2, 1. The correct answer is C. Proteins may be excreted/secreted from the cell. Let's continue on. What is the cytoskeleton? Our cytoskeleton is used to give the cell shape. You can think of the cytoskeleton as the filaments that organize the shape of the cell membrane/plasma membrane. And there are three main filaments within the cytoskeleton. We have microtubules, actin filaments, and intermediate filaments. The big guys are the microtubules, the tiny ones are the actin filaments, and the in-betweeny ones are the intermediate filaments. As we look at some extensions that come from the cell membrane and part of the cytoskeleton, we have cilia and flagella. Both of these extensions have microtubules, so those large tubes from the cell cytoskeleton in them. And we can use these for movement. A key difference is the flagella moves the entire cell within the human being. Here's a sperm cell with a flagellum. Notice how big the flagellum is in comparison to the cell body. And the cilia are much smaller than the cell body. Cilia are about 1/20th the length of a flagellum. And the cilia do not move the cells in human beings. In human beings, cilia move substances past the surface of the cell. So in human beings, flagella move cells, and then cilia move substances past cells. That's an important distinction. We also have some special connections that hold our cells together. These cell junctions are going to cause two cells to interact with each other in different ways. We use cell junctions to hold cells together to make tissues. And to review, a tissue is made of two or more cell types working together for a common purpose. The three main kinds of junctions include adhesion junctions, gap junctions, and tight junctions. So let's first talk about adhesion junctions. Our adhesion junctions are going to mechanically attach cells to each other, and these adhesion junctions are nice because they allow for lots of stretching and bending of the tissue, which is one reason why they are common in skin cells. We also have tight junctions. Tight junctions cause the plasma membranes of cells to be directly linked to each other. Because these tight junctions directly link plasma membranes to each other, as opposed to the adhesion junctions, which allow for a gap in between the plasma membranes, these tight junctions form a waterproof barrier. So they're very common in the digestive system, where we don't want our digestive enzymes to get out and digest the rest of our body, or our kidneys, where we need to have lots of fluids being isolated from each other. And then finally we have gap junctions. Gap junctions are unique in that they allow chemicals from one cell to be transported directly to the next cell. You can think of them as a small tube that connects the cytoplasm of cell A to the next cell, cell B. We typically are going to use gap junctions when it's important to have chemicals move directly from the inside of one cell to directly to the inside of the next cell. Gap junctions are very common in cardiac muscle tissue, the muscle tissue of our heart, which allows for electrical impulses to move efficiently from one muscle cell to the next muscle cell within the heart. So, concept check class. Which of the following cell-to-cell junctions form a waterproof barrier? Gap junction, tight junctions, adhesion junctions, or all of the above? Go ahead and rewind the video or pause and check your notes / textbook and get me an answer. 1, 2, 3, 4, 5. The correct answer is tight junctions form the waterproof barrier. Gap junctions form little tubes that connect the cytoplasms of cells. Tight junctions form the waterproof barriers between cells, and adhesion junctions form flexible barriers between cells. Let's continue on with metabolism of the cell. The metabolism, generally speaking, is all chemical reactions in the cell. And this is different from how we typically use the term metabolism in everyday speech. Typically, in everyday speech, when we use the term metabolism, we are going to say that we're talking about somebody who burns a lot of calories without working very hard. Yeah, we all wish we had that kind of metabolism, right? But in this context, metabolism is any chemical reaction in the cell, either breaking up down a molecule or building up a molecule. Generally speaking, in our cell, all these chemical reactions are going to start off with products that go into the chemical reaction and then act as a reactant. Reactants that go into the chemical reaction and get turned into products. I had that flip-flop there. Sorry about that. So here, if we look at chemical reaction number 1, A is the reactant, and then B gets turned into B, the product. For chemical reaction number 2, B is the reactant and gets turned into C, the product. What we can see here is a metabolic pathway. We have 6 chemical reactions where the reactant for one chemical reaction is the product for the other chemical reactions. If we look at chemical E right here, chemical E is the product of chemical reaction number 4 and a reactant in chemical reaction number 5. Chemical E was made by reaction number 4 and goes into reaction number 5. Products are made by the chemical reaction, reactants go into the chemical reaction. As we're regulating these chemical reactions, generally speaking, we need some kind of enzyme. An enzyme is a chemical or a substance that speeds up a chemical reaction. And the vast majority of all enzymes in our cells are made of proteins. Enzymes are typically going to be named for the molecules that they act on, or the substrate molecule. And it will have an ASE at the end. So a lipase, Lipase, is an enzyme that acts on lipids, or a sucrase is an enzyme that acts on sucrose. These enzymes are going to be very specific as to what specific substrate they can work on. All enzymes are going to have a spot on their molecule that the substrate attaches to. This spot is known as the active site. And another key point for our enzymes is that enzymes are not going to be used up in the chemical reaction. They can be recycled and used over and over and over again. Also, to speed up our chemical reactions, our enzymes are going to make the chemical reaction more energetically possible. And this is kind of a technical term right here. Energy of activation is the amount of energy that is needed to start the chemical reaction. A good example of energy of activation is setting a match on fire. Matches all by themselves, sitting there, generally speaking, do not burst into flame. But if you apply a little bit of heat to the match by striking it on the sandpaper of the box, that heat provides the energy of activation needed to ignite the match. When enzymes lower the amount of energy needed to start the chemical reaction, when they lower the energy of activation, they make it easier for the reaction to take place, and the reaction takes place quicker or at an accelerated rate. Also, some enzymes are going to need a non-protein helper. Those non-protein helpers are vitamins and minerals that can be referred to as coenzymes and cofactors. So let's look at some pictures, because boy, was there a lot of words on this slide. So now let's look at this in a picture form. We can have a substrate, the chemical going into the chemical reaction, attached to the enzyme. And specifically, the substrate attaches to the active site on the enzyme. When the substrate and the enzyme are attached to each other, they form what is known as the enzyme-substrate complex. And then the substrate, it reacts with the enzyme, or I should say reacts in the enzyme, and the substrate gets turned into products, and then the enzyme is unchanged and can be reused all over again. Sometimes enzymes can break down molecules, so we can start with a big molecule and end up with a big substrate and end with smaller products. Sometimes we start with small substrates and end with bigger products. So we can have a breakdown reaction or a synthesis reaction. These can also be referred to as catabolism or catabolic reactions and anabolic reactions, to use some more technical jargon. If we look at the energy of activation for an enzyme, on the y-axis of this graph we have the amount of energy required, and then we have time, or the progress of reaction. So here on the left side we have the reactant, the unreacted substrate or reactant. Here at the other end we have the energy of the product. So the chemicals going in always have the same amount of energy. The chemicals going out always end with the same amount of energy. The difference is how much do we need to increase the energy for the reaction to take place. Without an enzyme, you typically need to add more energy for the reaction to happen, but with an enzyme you add less energy. You need less heat or less ATP energy for the reaction to take place. Let's continue on and look at our mitochondria. Mitochondria are the power plants of the cell. They make energy in the form of adenosine triphosphate, that ATP we had talked about. Mitochondria are a double-membraned organelle. They are one of 3 double-membraned organelles within the cells, and they are going to be present only in eukaryotic cells. The inner membrane of the mitochondria has many folds on it, and these many folds increase the surface area so that we can maximize the rate of reaction and the rate of ATP production within the mitochondria. Current theories suggest that the mitochondria was initially a separate prokaryotic cell that was engulfed and became part of the eukaryotic cell. Some evidence that supports this is the fact that mitochondria have their own unique genomes and their own ribosomes on the inside of the cell. So these little dots right here represent mitochondrial ribosomes. These are made exclusively in the mitochondria and stay in the mitochondria, and the mitochondria have their own genome as well. That is something that is quite exciting within the field of micro and cellular biology. Next, we're going to talk about cellular respiration, and this is a topic that many students find quite intimidating, particularly if you take human physiology. We spend lots of time grinding through cellular respiration. In this class in human biology, we're going to be kind of briefly skimming over the highlights of cellular respiration. In general, cellular respiration is a term that refers to how we make ATP, adenosine triphosphate, within the cell. And there are 3 main steps for cellular respiration. We have glycolysis, the citric acid cycle, which is formerly known as the Krebs cycle. This is a really old school term. I'm going to be using the term citric acid cycle. And then we also have the electron transport chain. And these 3 steps work together to make most of the ATP. And because we've already talked about how the mitochondria makes most of our ATP, you can imagine that most of cellular respiration takes place within a mitochondria. To summarize everything for cellular respiration, we'll start off with our bloodstream. And our bloodstream is going to have oxygen and blood sugar go to the outside of the cell, the interstitial fluid. That oxygen can be absorbed into the cell. That blood sugar can also be carried into the cell. In the cytoplasm of the cell, we have glycolysis take place, and then in the mitochondria, we have the citric acid cycle and the electron transport chain. That oxygen is going to go into the mitochondria to work in the electron transport chain. That glucose goes into glycolysis. The glucose gets turned into pyruvate. The pyruvate's then sent to the the citric acid cycle, where electrons are ripped off and sent to the electron transport chain. Glycolysis makes 2 ATP. The citric acid cycle makes 2 ATP, and the electron transport chain makes 32 ATP. And these numbers are all per glucose molecule. So 2 ATP per glucose, 2 ATP per glucose, 32 ATP per glucose. So cellular respiration with oxygen produces a total of 2 plus 2 plus 32, 36 total ATP per glucose via cellular respiration. Let's talk about glycolysis in a little bit more detail. It's always going to occur in the cytoplasm of the cell. During glycolysis, 1 glucose molecule is broken into 2 pyruvates. So this 1 glucose right here is turned into 2 pyruvate molecules. NADH is an electron carrier, and we are able to make NADHs and pick up high-energy electrons and carry them away from glycolysis. Glycolysis also makes a total of 2, or net yield of 2 ATPs for every 1 glucose. Something else that's worth emphasizing is glycolysis does not require oxygen. So if you hold your breath you can have plenty of glycolysis taking place, and you can make those 2 ATP over and over and over again via glycolysis. Our citric acid cycle is a cyclical pathway. So we go back here, we can see the citric acid cycle is represented by that circle. And it's going to make many NADHs. Most of the high energy electrons, the NADHs, are going to be produced in that citric acid cycle. And we also make 2 ATP from the citric acid cycle as well. The NADHs made by glycolysis and the citric acid cycle carry their high energy electrons to the electron transport chain. Something that is also notable about the citric acid cycle is this is where the carbon dioxide that we exhale is produced. So when you're working really hard and you're breathing really hard and making lots of carbon dioxide, that carbon dioxide is made in this chemical pathway, the citric acid cycle, inside of the mitochondria of our cells. Finally, all of the NADH that's made in the glycolysis and in the citric acid cycle are going to carry their high energy electrons to the electron transport chain. This electron transport chain takes place inside of the mitochondria on that inner membrane. So we can see those folds on the inside of the mitochondria right here. That is the inner membrane of the mitochondria where the electron transport chain takes place. Depending on the kind of cell, we can make 32 ATP in the electron transport chain or up to 34 ATP on the electron transport chain. Something that's notable about the electron transport chain is you need to have an electron acceptor at the end, a final electron acceptor. For us, for human beings, our final electron acceptor is oxygen. So that's why we need to inhale oxygen, so that we can have those oxygens in the electron transport chain to act as the receiving, or the final receiver or receptor of the high energy electrons that have been ripped out of our glucose from glycolysis and the citric acid cycle. Now, that was a very brief overview of the cellular respiration process. It's worth noting that we can also make ATP out of other kinds of carbohydrates like starch or sucrose or fructose. We can also make ATP out of proteins that we eat, and we can make ATP out of lipids or fats that we eat. Depending on what we eat, these different carbs, proteins or lipids, can be plugged in at different spots. Sometimes those different carbohydrates are turned into glucose and funneled all the way through. Sometimes the proteins or the lipids are plugged in into the citric acid cycle to help us make ATP. Now, we also have the ability to make ATP without oxygen present. This is known as a fermentation pathway, and it occurs exclusively in the cytoplasm of the cell. Fermentation, or anaerobic without oxygen respiration, does not require oxygen. It's going to involve glycolysis because glycolysis doesn't need any oxygen. We'll break down a glucose into pyruvates. During the process, we make 2 ATP, and then instead of sending those pyruvates into the mitochondria, we're going to take those pyruvates and turn them into a lactate molecule or a lactic acid molecule. And this can be used to give us a quick burst of energy when we're in a short or low oxygen environment, but long term we cannot sustain fermentation. Fermentation only produces 2 ATP from glycolysis as opposed to the 36 ATP that we get per glucose that we're used to. So if we're in a bind, we can do glycolysis for fermentation for a short period of time, but long term we need to go back to aerobic respiration or typical cellular respiration because we need the higher ATP counts. So, let's see here. Check. Concept check class. Which part of cellular metabolism makes the most ATP? Is it fermentation, glycolysis, the citric acid cycle, or the electron transport chain? Go ahead and rewind the video, pause the video, or you can pause the video, check your notes, or check your textbook. Please get me an answer. 1, 2, 3, 4, 5. Class, the correct answer is D, the electron transport chain. The electron transport chain produces 32 to 34 ATP molecules. Fermentation produces 2 ATPs. Glycolysis produces 2, and the citric acid cycle produces 2 ATPs. So the correct answer is D, electron transport chain. And that's all we have for our presentation on cells. If you have any questions about the information in this presentation, please feel free to ask me while you're on campus. You can also shoot me an email, or you can post them in the class discussion boards. Happy studies!